All right, 9:30, let's begin. Okay, so good morning, everyone. Let me switch the lights down. Yep. So, 2 lectures. One, it's going to be on mycology and parasitology in general, so a little bit of an overview. And the second hour I like to spend on Plasmodium and the disease that is associated with an infection in people, malaria. And this is a kind of special attention I previously gave like important viruses, and that is because malaria is an ongoing problem. Not so much for Australia, but for many parts of the world. And Australia is at risk of getting endemic malaria all the time if they do not keep the guard up. So it is important for us too. And this is also reflected in that some of the best institutes doing research on malaria are in Australia. So there is clearly a focus on this. Okay, but start with fungi and with parasites first. So the information here on fungi is a little scattered over our textbook. So here are some pointers, but I'm sure you can work the index and find even more. And there is, of course, also further information on parasites. So let's start with the basics. So to the left, we have a fungal cell, in this case a yeast cell, and to the right we have a kind of a typical general description or representation of a protozoan cell. So, and as you can see, these are very similar, but similar does not mean identical. And again, we have to find the differences, in this case the differences of fungal cells to protozoan cells or to our cells, if you ever want to specifically target them with drugs that inhibit the multiplication of fungi but don't kill the host. So, and one really point of difference is that all fungi have a cell wall. So we, our cells and protozoan cells, do not have a cell wall, but fungi have a cell wall. Plants also have a cell wall, and this in some aspects is similar. The fungi, however, have a cell wall that is made of chitin. And here we have a few more basic facts. So we have the fungi here in this column, and then we have algae, protozoa, and helminths. And maybe I would like to say a little bit about the cellularity. So yeast, that's the stuff you use to make bread or beer. They are multicellular. And the yeasts are unicellular, so you have individual cells, whereas all other fungi, so for example, the fruiting bodies you see in the forest, they are multicellular. Okay, so previously I had a whole portion on these multicellular fungi, but because they are not all of medical importance, so we skipped that for having a bigger focus on medical science. So, but let's dwell a little bit on the cell wall and on chitin. So, the chitin is not restricted to fungi. You may already know that you find chitin in all of these critters, so in spiders, in insects, in centipedes and millipedes, in crustaceans. And what I learned rather recently is not even restricted to this. So there are whole other groups, so for example bryozoa, who can also make chitin. So chitin is relatively widely distributed, but in this case for us it's important because we do not make chitin, but the fungi do, and it also makes the fungi rather resistant to environmental stress because chitin is a very durable biopolymer. So it's a polysaccharide. It consists of unit of acetylglucosamine, which are linked in a beta-1,4 fashion, that's depicted here. And this is all very similar to glucose in cellulose. However, they have acetylamine group in addition, and that allows for interactions between different chains, and that gives the whole cell wall an increased strength if you compare it to the cell wall that consists of cellulose in plants. So, and fungi, many fungi are pathogens, and some of them are pathogens in people. So, and if you have an infection that causes you problems, then you suffer mycosis. And fungi can cause severe infections. So we previously came across Candida albicans and Pneumocystis jirovecii or X. carinii, just if you Google this and you look this up. So this is not a confusion of my part. So this thing has been renamed. And this happens from time to time. It's nothing really unique. And they also, I think also mentioned that some penicillium species are a real killer amongst people with HIV/AIDS. So what you can see here already at this small selection of pathogens is that sometimes you have like pathogens like Candida, who we all come into contact earlier or later in our life, or almost all, but it does not really kill us, and in most of us doesn't even cause a problem. However, under certain circumstances, this can be a severe problem, and this is then really imminent and important if it comes to severe immune suppressions. Okay, so what most people at one point will see is something like this, and that is sometimes also called an athlete's foot, at least in English. And it's not always for people who run, so even couch potatoes can do it, I think. It's a weird thing here. And that's a fungal infection of the skin, so which is a bit uncharacterized. So it's not like a description that is specific for an infection with one fungal species. So it's basically a fungal infection between your toes. And it sits there and infects that preferentially because there's an ideal environment, because it's humid and warm. And that really describes very well what the fungi like. Okay, and now something historical. And so fungi as pathogens in animals, and fungi as, and the link to early pregnancy tests. So you may have heard, and there was something in the news recently on the efforts of people at this university to fight the citric fungus in amphibians. And that is a thing that kills many frogs, and actually kills many frog species as such around the globe. And that is a fungal infection of the skin, and that has recently spread all over the globe and kind of wipes out entire populations. And that is a citrid fungus, and citritomycosis is a disease, and this is a particular species that does it. So then you may wonder, so why did that disease spread so efficiently around the globe? And some people believe that has something to do with the popularity of the Xenopus frog some decades ago as a means to do pregnancy testing. So you have now all tested in the lab a urine sample for a hormone. So that's how modern pregnancy tests work. But before that was developed, so they had a rather crude test. So they took Xenopus frogs that were injected with a sample of urine from a pregnant woman, or a woman supposed to be pregnant. And if the frog ovulates and laid eggs 12 hours after injection, the woman was deemed to be pregnant. So this test did work well, but you can imagine that that was much more expensive than the rat test we gave you, and you can buy at the pharmacy if you want to perform such a test. And it also required skill, whereas to pee on a stick everyone can do this, but to have like frogs ready and to inject them and to monitor them, that's all kind of high effort medicine, if you like. So, but that means that these xenoprus frogs were shipped all over the world for pregnancy testing, and with them a fungus that isn't actually that pathogenic for the xenoprus frogs, but then the xenoprus frogs ended up as pets and maybe undiscarded ones in the wild, and also maybe not everything that came out of the laboratory was probably decontaminated and sterilized, so this may have aided the spread of the fungus. So this is possibly, as far as the amphibians are concerned, a rather negative effect of modern, or as you see now, outdated medicine, but I think from a historical perspective, probably worthwhile noting. Okay, so now we come back to Pneumocystis and it being so deadly in immunocompromised individuals. And there's also a historical note. So this is the one where the species name was changed recently, and this is the one I told you led to the discovery of the AIDS pandemic in North America, because physicians and the CDC, when the CDC was led by intelligent people, they were very smart in picking up an unusual spike in the use of the antibiotic pentamidine. And that was used to treat these infections. But normally that was super, super, super rare. So then they worked backwards and basically found that a disease was spreading that would suppress the immune system. And then, with the help of the French who isolated the virus first, then the correlation between HIV and AIDS was discovered. So this fungal disease was basically instrumental in the discovery of HIV/AIDS. And what is also historically interesting is that this particular pathogen and many others were previously deemed to be protozoans, not fungi. So because they looked and behaved like protozoans rather than fungi. And however, with the advent of sequence analysis, this was not really tenable anymore. So therefore that was reclassified, and not just, we got the species classification in the genus wrong, so that was on a much higher taxonomic level. And historically it's quite remarkable, and you can still hear some echoes now, that they will say, DNA is just one argument amongst many others, but we have all this structural evidence. To be frank, this is all bullshit. So in this case, DNA basically trumps all other arguments. But it seems that it needed a generation to have that shift done. Okay, now we come to antiviral drugs. Amphotericin B was discovered in 1955, is still used today. Is not without side effects. ACE is not something that is easily prescribed, but it is something that really works. So it's naturally produced by bacteria, not for us, but in an effort to wage war amongst other microbes. So that's what it always is here. And it can inhibit a wide range of fungi. However, as I mentioned, there are severe side effects. So it works by binding ergosterol, and that is something that exists in the cell membrane of fungi, but not in our cell wall. So that makes it somewhat typical and specific for fighting fungi. And it forms pores in the cell wall. And that results in the leakage of many ions, and that eventually kills the fungus. Note that bacteria do not use sterols in the membranes and therefore are not susceptible to these drugs, which makes sense because it's produced by bacteria. So then at least this particular bacteria must be resistant, otherwise it would kill itself. So here is a little bit of a depiction of that, and it's sometimes called a death ring, and it's kind of a polymerization event that leads to a pore that spans the width of the membrane and then generates that leakage. Okay, there are other fungal drugs. Clotrimazole is an example. This is quite hilarious because, I don't know, this is some early influenza picture, but you are not supposed to smear it on like this. So fungi are often, and I'm not an exception here, a little treated on the periphery only, and we have just one lecture on it. But I would like to see like a more dedicated, maybe a whole lecture series of fungi. I think there is a whole new world out there, not just as parasites. But we, however, have to move on a little bit. Plus, I'm not really the expert in this. Okay, so that's my little crash course and introduction in the world of fungi. So, and you also see, if you see the textbook, it's also more focused on bacterial infection and virus infection than it is on fungal infections. And there is not really a pandemic that is based on, or that goes back on fungal infections. Maybe that's why. Okay, we shift now to parasites, and we have a few definitions to take up first. So, parasites obtain food and shelter from other organisms. That's the very definition of it. And we have obligate parasites, and their survival depends on a host. So they will not survive individually and as a species without a host. So also that means if you kill the host, if you can, then you will also kill the parasites. Or if you kill one host they have to go through, you will also kill the parasite. And that is a strategy that is actually used in some cases. Then we have facultative parasites. They prefer to live in hosts, but they can also survive and multiply outside, and they are a little harder to manage. Then we have endoparasites. As the name suggests, they live inside the body. Ectoparasites on the surface. Pathogenic parasites, they cause disease. Other ones, the definition blurs a little bit at the edges. So we have many organisms that live with us but cause minimal harm or no harm, and when something starts to become a parasite and not just a comrade, if you like, that's sometimes a matter of perspective. In medical parasitology, the term parasite has come to mean a eukaryotic pathogenic organism. Thus, protozoan and metazoan infectious agents are classified as parasites, while bacteria and viruses are not. The basis for this escapes me, but this is how most books, including our books, handle the matter, so you may as well be aware of it. Interestingly, fungi are not discussed in most textbooks of medical parasitology, even though they are eukaryotic. So it's not even very consistent, but this is all not my definition, so these definitions are used. So whether we like it or not, we should be better aware of it. Parasites can be unicellular or multicellular, and they can be so big that the length of the parasite can actually be longer than the length of a host. So we can host tapeworms that are considerably longer than we are tall, for example. So they don't have to be small. Host definitions. So we have definite host, and this is a host in which the parasite reaches maturity and, if applicable, reproduces sexually. Then we have intermediate hosts, and that's a host that harvests the parasite only for a short transition period, during which some developmental stage is completed. Asexual reproduction may occur. But that doesn't mean that the intermediate host can be skipped or is unimportant. It's just the host where the sexual reproduction does not occur. Then we have a reservoir host, animals that host a human pathogen while it isn't infecting humans, and are used by the parasites as sort of maintenance. So then we have a vector, and that transmit parasites, and this is also a host, and we will see this in quite some detail when we look at Plasmodium in the next lecture. And then we had dead-end hosts. It's always hard to avoid a pun here. And this is an intermediate host that does generally not allow transmission to the definite host. thereby preventing the parasite from completing its development. For example, humans are dead-end hosts for canine tapeworms. As infected humans are not usually eaten by dogs, foxes, etc., the immature tapeworm, although it causes serious disease in humans and can actually kill its host, is unable to infect its primary host immature. So the error of this particular parasite may result in the death of the host and the death of the parasite without the parasite having a chance to, live on. So that's a dead-end host from the point of parasite view. Okay, so parasitism is a very successful lifestyle. So there are more parasitic than non-parasitic organisms on our planet. And if I do a little paleontology, I always have a keen eye on fossil parasites, which gives you a little edge and makes you selling otherwise rather boring stories. Okay, but now we go back to important parasites in Australia. The good news is that the most devastating parasitic diseases of humans, and that includes malaria, sleeping sickness, and schistosomiasis, are not endemic in Australia. The bad news is that parasitic infections are frequently found in remote Aboriginal communities. I've actually seen it. It's not so remote, and this is a little bit of a disgrace to the entire nation, in my opinion, and it's a bit beyond me why this continues to occur. Parasites are a significant burden to Australia's meat, wool, dairy, and aquacultural industries, so there's money to be made in keeping the parasites at bay. And the examples are listed here and in the following, rather than reading them all out. I will give you some slides on some of those. Okay, let's start with sleeping sickness. Again, it is nothing you find in Australia. So there are two clinical forms. One is slowly developing and one is rapidly developing, and they're caused by different subspecies of Trypanosoma brucei, and they have a preferential occurrence either in West or East Africa. And in the past, the disease killed many people, but thanks to medicine, since the late 1960s, it really dropped dramatically. And in recent years, we were about 1,000 per year. So eradication seems possible, but sometimes the WHO struggles with the remaining cases. And that's a little hard, because then you have to keep up this effort for longer than making a real effort and giving everyone on board and that's solving problems once and for all. So we have seen this, for example, with polio, and we've seen this with measles. And that's why it makes me so angry that people who should know better, because they're educated, act against it. And the American current leadership is trying very, very hard to bring measles back to the United States. But to get infected with measles, you do not have to travel to the United States. You can also do this in certain areas in Australia where people congregate that refuse to vaccinate, and then you have circulating things like measles. So, okay, but coming back after this rant to sleeping sickness is not in Australia and it's not likely to be in Australia. Okay, so, but there is something why I got on the sleeping sickness and its vectors and the agent that causes it, because there is something very important in connection with immunology. So this is a parasite, and this is a smear of blood. And what you see here is a bunch of erythrocytes, so these are your red blood cells. And they do not have a nucleus, so they look like very boring, because there is almost no structure to see with a microscope like this. But you clearly see the parasites. And the parasite in this particular case, in contrast to the infection of people with Plasmodium, the parasites live outside the red blood cells, not inside cells. Okay. This is a bite reaction that occurs when the tsetse fly who transmits the disease has bitten a human, and And this is a Winterbottom sign. And these are swollen lymph nodes in a child with early trypanosomiasis. And this is the late or the final stage when now treatment is administered. So one should not see this anymore because it can be treated. Why trypanosomes are so effective in overwhelming the immune response in people, untreated people that is because they have developed a very fancy strategy to do so. So they are equipped with what's called VSGs, variable surface glycoproteins. And these are glycosylated proteins, so proteins with sugar chains on the surface of the parasite. And this is basically like a coat. And that enables the parasite to evade the host immune system, allowing chronic infection. The true properties of the VSG coat that allow immune evasion are a shielding the dense nature of these sugar molecules, prevent the immune system of the mammalian host from accessing the plasma membrane and invariant surface epitopes, so those that hardly change, because if you would change them, then you would change the function and the parasite would suffer. And they are kind of hidden in that forest of sugar chains. And this wouldn't be so much of a problem because eventually the immune system would learn to make antibodies against these terminal sugar molecules on these chains, so as we have antibodies against all the blood groups. So that's possible. However, it doesn't happen, and that is because of periodic antigenic variation. The VSG coat undergoes frequent genetic modification, it's called switching in this case, which leads to variants expressing new coat proteins and new sugar chains. This is instrumental to escape the adaptive immune responses mediated by antibodies and T cells that have been raised against the previous coat. So, if a person is infected with trypanosoma, and here this is depicted here with the parasite depicted in red, and this is timeline weeks after infection, and this is a relative number of parasites in blood. So after infections, the number goes exponentially up, and then at some point peaks and then goes down. And it goes down because the immune system gains the upper hand, and antibodies are made, and T cells are directed, and then all these parasites are killed. So, and that would normally be the end of it. But then this clone appears, which is depicted green. It's not green, it's just to distinguish it in this graph from the previous one. And this manages to have another exponential growth curve. And that is because one parasite of that clone has switched to different genes that make a different, that encode different code proteins and then provide a different code, and looks totally different from the previous one. And all the antibodies that were produced against clone A are now useless to clone B. It's like a new infection. And then this one had its time under the sun, uncompromised by immune responses, and then the immune system catches up, makes antibodies against clone B. But because constantly some members of clone B switch to even other genes that code for different code proteins, then a new clone appears and the whole thing happens again. And this can happen because the genome of trypanosomes, it's very complicated. So they have 11 pairs of large, 3 to 5 intermediate, and 100 mini chromosomes, many, many more chromosomes than we do. And the small chromosomes carry many of the genes involved in antigenic variation. And they can all be exchanged through sexual reproduction, and it's all fluid and it's highly redundant. So they have a huge repertoire of genes to make different code proteins and basically change their appearance over time. And that helps them to cause a chronic infection in the face of an otherwise effective immune system. it's like, I don't know, does it exist in English? The story with the hedgehog and the hare, where they race, and they're actually two hedgehogs that just pop up in between the posts, and the hare runs, but never wins. Okay, maybe this metaphor goes a little too far, but by the end of the day, this is very hard to deal with by the immune system, and you can see, because it causes chronic infection that often leads to death. So, which is my example, and we will find another one when we discuss malaria, that these parasites face the immune system and they have to do something about it. This one lives in the open, in between the blood cells, and still has to evade the immune system, and it does so by going through a series of antigenic variations. Plasmodium that has a different story, as we will see in the next lecture. It hides inside the red blood cells, and it's only basically for seconds outside the red blood cells. So the host can make an immune system as much, or immune response as much as they want. Most of the time, the Plasmodium parasites are not even exposed to this. So there are different ways to deal with the problem from the parasite's point of view, and they can all be successful, as we see, and it's not one is better than the other. And this is a fine example for having this rapid evolution inside a host, in every single host, at all times, and very successful. And also, if you ever had trouble believing in evolution, you can see it. This is very fast. This happens within the lifetime, even if shortened, of a single person. Okay, so much to trypanosoma. Cryptosporidiosis is the most common non-viral cause of diarrhea worldwide, so can have quite an impact. When ingested, sporozoites come out of the oocyst and invade the epithelial cells of the intestine. Cryptosporidium is a problem for immunocompromised patients. People can literally die of diarrhea by losing over 13 liters of fluid a day. This is enormous. You can drink all the time. It will not make up for it. So this is very serious. Cryptosporidium is highly resistant to the levels of chlorine normally found in drinking water and swimming pools. There are no real effective drugs to treat the parasite. Because of its small size and because it is rather resistant to disinfectants, Cryptosporidium is a serious problem for the water industry. So there have been a few very well documented, investigated and publicized cases. So famously, there was one in Wisconsin in 1993, where 400,000 people became infected. So this is really something serious. However, close to us, this also sometimes causes concerns here. So here we have ABC News, nearly 500 cases of cryptosporidiosis recorded in New South Wales this year as Health Department issues alert. So everyone needs to be alert, not just an issue of the past. So this is ongoing. Okay, giardiasis. I like them because they look really spacey, as we see. So we see the parasite here. It's characterized by a huge ventral sucker that allows them to stay in the small intestine where they live. And here we have the life cycle, but I do not really want to go through every single life cycle of every parasite. That is something for you to look at home. It's also a common cause of gastroenteritis. Infects approximately 200 million people worldwide, so it's a big thing. It includes loss of appetite, lethargy, fever, explosive diarrhea, stomach problems, bloating, and flatulence. Symptoms typically begin 1 to 2 weeks after infection, may wane or reappear clinically. Most people are asymptomatic. Only a third of infected people exhibit symptoms. And this is also something you can get if you run out of water and drink out of, dams and streams and things like this. So, if you're a hiker, you should make sure that you find a way to disinfect your drinking water. Another one is toxoplasmosis. We came across this one when we discussed a little bit opportunistic infections in HIV-infected people with AIDS. So this is the one that normally kind of cycles between rodents and cats, but can also infect people. And this is the one that is supposed to have evidence is weak, but the story is good, which makes it very difficult to get rid of. That there might be scope for more research, I'm saying, is that it could be a mind-altering parasite. Interestingly, some parasites have quite complex and intricate behavioral changes, can cause these in their hosts. You may have seen that infected caterpillars, before the parasites leave, they go to certain places, or that ants do not go back to their home, but bite into grass and stay overnight outside in the hope they were eaten by the following host in the morning. So these things do happen, and this is supposed to make mice a little fearless, and then increases the chances to be eaten by cats, and that promotes the change of a host for this parasite. Okay, now we go to helminths. So these are sometimes called worms, but worm, I cringe a little bit, because worm describes the shape of a thing rather than, a particular taxon, because things that look like worms are not necessarily closely related. But many of these things look like worms, so I can give you this. So, and helminths are very important pathogens of people, and you see here the number of people infected in 100 millions, so this is 100 million. So you see that all of those are quite considerable. That does not mean you have a fair chance of getting them in Canberra. You probably don't, but if you go to other places in the world, you still get those. And which brings me to ectoparasites. This, however, is something you can very well get in Canberra, which puzzles me a little bit. Not to say that in other cities of the world you will be better off, but this is something that seems to be really, really hard to kill. So what we see here is a cam, and this one is, I think, does it say somewhere, 6th century BC. So that's 2,600 years old. And this one you can still buy at Coles. So if you look at it, nothing has changed. And why people use these devices is because they want to get those, or rather with the fine side of it, and you can see that the old early example is exactly the same, can rid of those. And these are nits, and these are the eggs that are laid on hairs, and out of which, the egg capsules rather, we have then the adult lice. So we have two of them, not all the time. One is the head or body lice, and the other one, or the head or body louse, and the other one is the pubic louse. So the pubic louse lives down there, and the head and body lice, they live here. And they look a little different, and they're actually finely adapted to the thickness of the hair. So the claws, which they use to hold on to the hairs, are a little more petit for these hairs, and they're a little more rough for those hairs. So, and you can also see that pubic louse is basically sexually transmitted, and head and body lice is very easily transmitted without sexual contact. And in fact, it's near impossible to have kids without them once coming back with a notification saying, oh yeah, there has been an outbreak of lice at school, and then you eventually try shampoos, and I can tell you, you will end up with these combs, or cut the hair off more or less altogether. So these things still exist, And it's amazing that it's probably here to stay for a while. So this is just an oddity about fleas. So, but what makes fleas apart from they are bloodsuckers, and you know it's a bit painful if you are bitten, and if it gets infected later on, then you may have even more trouble. In the past, fleas transmitted very devastating diseases, and that eventually was a lot worse than corona. So, for example, the bubonic plague. And this goes back to a bacterial infection that is transmitted by fleas, at least some forms of it. So if you look at the plague and you sometimes see, okay, there's a qualifier. This is a bubonic plague, a septicemic, or pneumonic. And bubonic is the least deadly, and that you get when you get bitten by a flea that is infected with the bacteria. Septicemic is infection of the blood and more deadly than the bubonic plague, because that is a kind of a widespread of the bacteria in your body, and it really becomes dramatic when you get a lung infection. And that is then almost always fatal. And that is also very contagious, because if it reaches the lungs, it does no longer rely on fleas, but then people can give the plague to each other without the need of fleas, because they cough it up and then the others inhale it. And then you kind of bypass the vector, and then it really goes. And this has led, and by the way, this is the bacterium in question, Yersinia pestis, and this has led to a loss of 30% of the European population in the Middle Ages, which had all kinds of consequences. It was obviously very bad for the 30% who died. It could also be bad for people who survived, but it also could provide a big chance. Because of this, it is alleged, at least as you know, my own history lessons go, that the cities were very desperate for new citizens, because you know they lost 30% of the citizens. So they offered free citizenship. So if you made it to the city and were in the city walls, you could escape your lord and you would be free. And this inspired then a kind of a revolution and independence thinking and all of this. So it basically moved civilization. At the course of it might have been the Black Death. But still, as a 30% is a very high price to pay for it. Okay, global distribution of plague outbreaks, and we are now not talking the Middle Ages. We are talking now. And what you see here marked in red are areas where you may get infected with Yersinia pestis bacteria, and you personally get to experience what the people in Europe experienced in the Middle Ages. So, and in the past, 100 years ago, there would have been red spots in Australia. So this is not completely dealt with. And some people who are lovers of exotic pets and animals, I think the prairie hound or something in America is a frequent host of that particular bacterial species without being much compromised by it, but it can happily pass it on to people. So it may not always be a good idea to have exotic pets. Yeah, here is the story about Australia. Between 1900 and 1910, Sydney, of all places, recorded a total of 1,371 cases and more than 500 deaths. And back in the time, they had, like, I think it was called shanty towns, and it was basically slum-like living conditions. And this was then the trigger to clean this up. And that was also in correlation with heavy manifestation of rats and fleas. And the plague, 100 years ago in Australia. again, something that should not really have happened. But okay, so it's not happening now, so Australia doesn't have a spot anymore. Okay, that brings me, after 45 minutes, to this image here. And what you see here are red blood cells, so this is one and this is one. And these here, they look a little different, but they are also red blood cells, but they have suffered a lot because they have been abused by the Plasmodium parasite to produce offspring, and this is just coming out, and this is colored here in yellow. And this brings me to the learning objectives for this lecture, and then to the end of it, 2 questions, to a 5-minute break, and then to the next lecture on malaria. So, questions first. That was a tour de force, not France, tour de force, through parasites. Is there anything you'd like to ask? Anything on the past lectures which I could not deliver face-to-face, but I'm happily taking questions to those as well. Yes?
Do all parasites actually reproduce inside their host or however they best exist, or does some asexually reproduce?
This is a question that can never really, because it's so diverse. So if you have the obligate parasites, yes, then they depend on the host for almost everything. Yeah, so, and then if you have parasites that like to live in a host, but if there's no host, then they just live in the soil, yeah, then they can do everything without ever needing a host. But they can do better in a host, that's why they go there. Yes. Yep. Yep.
Why is it challenging to develop drugs against fungi and parasites?
I'm not saying it's impossible. So the examples show that it has been done successfully, but it's possibly like on a, you step back and have like a bit of a bird's eye view, more challenging because they are closer to us. They are more similar to us. So these are cells that have a nucleus, they have mitochondria, like the overall organization is very close to our cell's overall organization. And that also means they basically share more ancestry with us, so their proteins on average are more similar to ours. So that means you have to work harder to find the differences. So that's what I meant to say. Yep. Okay, so if anyone wants to run to the bathroom, now is the time. And we start, I would say, in 5 minutes. Okay, so before we start on malaria, do you want to ask your question again to the benefit of all? So if I repeat this, is what kind of questions can we expect in the final exam? Right?
What kind of questions am I expecting?
Or topics, okay. Yes, so a final examination by definition should cover the entire content of a unit. So this is not my personal interpretation, this is how it should be done. Whether it's always done like this is a different story, but this is how it should be done. So, and this is how we will do it. However, so we have a practical skills assessment, how it's these days called, or a practical test that's coming up soon. And that will cover what I was taught in the practical classes. So, and the final exam, as I see it, will cover the stuff that is taught in the lectures and in the textbook. So, so for example, there will be no questions about API tests or cultivation media or things like that in the final exam. So this will all be dealt with in the practical test. However, we dealt, for example, with the cell wall of fungi and bacteria and gram staining in lectures and in the practicals. So this might be something that may happen in both, given the importance of the gram staining and the 50 times or so I mentioned it. This is maybe not even totally unlikely. But the focus, the very strong focus is on stuff that's the content of lectures. So in my opinion, if you know what are taught by learning the slides of the lectures, you should get an HD easily. Maybe you need the textbook to understand what to try to explain using these slides, but basically I design my questions by looking at my slides, not by looking at the textbook. Yes?
For the practical skills assessment, you know how usually in the manual there's an appendix with tables and stuff like that?
Yeah.
If that material is necessary to tables and stuff,
Then you will get it. So I cannot remember the stuff in the table, and I do not expect that my students remember the stuff in the table. That's what we have the tables for. And if you need a table, then you will get a table. So, and I think that's fair. But you will not get Wikipedia, and you will not get ChatGPT. So you have to learn something, but you do not have to learn which diameter of a zone of inhibition makes a particular species resistant or susceptible. If you have to do such a thing, then you will be given a table. Then you have to know how to use the table. Does that answer the question? Cool. Any other question? And if you have questions like this, yeah, I will come to your question. If you have questions like this, I pretty much prefer the questions to be asked in public and not in an email. I will be very short in my email responses or not response, because this is not an advice that should be given to individual students. It is advice that should be given to all students at the same time. Yes? Yes, I do, and I get into trouble if I tell you. Let's put it this way. I asked for the exam to be early in the 2-week examination period with the justification that my team has then time to mark it. Previously, I wasn't always happy with the examination office, so the current examination office seems to be a lot better than previous examination offices. So with that justification, I have a unit of more than 100 students, so there will be a lot of marking, and we need some time, and so I've asked for a very early time in that 2-week time period, and I think I will be given that time. Yes?
When will we find out when the exam is?
Again, I don't know. So these are the miracles of administration. So, because if I tell you exactly when it will be, and they shift it, then some students will blame me and say, I planned my life around the date you have released, and now I want to have a supplementary exam, and I definitely do not want to go there. Otherwise, I would happily tell you. Okay, any other question I can answer? Good. Malaria. Okay. There's a lot in the textbook on malaria, and this is all good, and I encourage everyone to read this and to read anything you can ever find on malaria, because I think it's an important topic. I have, however, some things in this lecture presentation that's not in the textbook. It doesn't mean it cannot be found elsewhere, and it doesn't mean it's not true, but occasionally I think I should present you and challenge you with things that aren't in the textbook. Otherwise, you can just buy the textbook and don't need me. So, come back to this photo because I think it's great, and I'm a stickler to electron microscopy photos, and I use this now to explain that in medieval Italians malaria means bad air, because they think that you catch the disease through the air, which is not totally unreasonable, because it's a vector transmitted disease, and the vector, and you should memorize this all, is a particular type of mosquito, and these are Anopheles mosquitoes, and only female Anopheles mosquitoes, and they transmit the disease, so it kind of flies through the air. Okay, malaria is caused by several Plasmodium species. So Plasmodium falciparum, and here we have the thing that then here thereon is abbreviated P. And here we have a couple of other species, Malaria, Ovale, and Vivax. And they all cause malaria, but by far the most important is Plasmodium falciparum. It's responsible for 80% of the infections and 90% of the deaths. So this is a real problem. There are even other related species that cause something similar in other animals. But these four are important to us, and therefore we stick for the rest of the lecture to those four. And this is from the textbook a generic representation of an apicomplexan organism. And Plasmodium is an apicomplexan organism. And they are called that way because they contain apicoplasts. And here I highlighted them. These are the two green things here. And this is another example for this kind of weird evolution where some cells have taken up other smaller cells and then turned them into organelles. So here we have the remnants of an ancient cyanobacterium. This is not to say that the apicomplexa can do photosynthesis, but they have the mitochondria, and they have something similar, and these are the apicomplexa. Okay. Geographic distribution. Now we don't talk about apicomplexa, we talk about Plasmodium falciparum and closely related species that give us malaria. So we see the north is fine now, and the south is fine. So the north was not always fine. So you could get malaria in Germany in the Middle Ages, and you could get malaria in Scandinavia at times in the Middle Ages. So that it is free now, it is because people did something about it. So, but you see that sub-Saharan Africa, and this line probably shouldn't be so sharp, that's where most of the action is, apart from parts of South America and, of course, Asia. A good thing to know is, because it has practical importance, is that if you go higher than 1,500 meters, you're fine. So you do not need to worry about it. So do you have any idea why this could be?
It's maybe less oxygen, like higher up.
And? No, mosquito doesn't like it. So no vector, no disease. So, and this is also one thing where we will see later, fighting the vector is fighting the spread of the disease in this instance. So, okay. But half the world's population are at risk of malaria. We are at risk, and there's a surveillance program in the north of Australia, and that's taken very, very seriously. So we could become endemic for malaria, but the fact that we are not endemic for malaria is because of constant effort. All right, so this is a distribution of malaria vectors. So these are different species of Anopheles. It's all Anopheles, as you see. There's a bewildering number of species. And you see this correlates obviously with the spread of the disease, but it goes beyond. And here you see that continental Europe, you have the vectors for the disease, but you do not have the disease. But if you do not have the vector, then you will never have the disease, because the disease depends in its spread on the vector. So this is an example. So this is a female mosquito, and you can see that the antennae are small. The antennae of the males are bigger because they want to sniff out the females. And the males do not really suck blood because they just want to copulate with the female and then they die. Whereas the females, they have to live on after copulation and to produce eggs. And to do so efficiently, they need food, and that's why they suck blood. And they suck blood to a point where they kind of are totally full, and to suck up even more, they have to get rid of fluid, and that's what these do. So that's a drop here, and that's a drop that had been dropped before. So to maximize protein intake. So they suck a lot of blood. So now we come to the general signs and symptoms of malaria infections, and there's a bit of a dot list here. So in general, it can be said that the disease can be flu-like and can resemble other conditions such as sepsis, gastroenteritis, and viral diseases, which I forgot the term that was used when I was taught this. So malaria is a great pretender. So it often gets misdiagnosed. So in particular, if people do not expect malaria. And this can fool experienced people. So I actually, as a student, missed out on microbiology. Before the first lecture was given, and that was just weeks before this, the lecturer died of malaria. So he actually does research that involves fieldwork in Africa, or did rather research that involved fieldwork in Africa, and attracted malaria, and that was misdiagnosed by him and all the doctors involved, and then he was dead before anyone could do anything about it. And they just saved his wife. Yeah, it was a bit tragedy. So this is serious, that's what I'm saying. Then what strikes most people is the intermittent fever, and that is, if that occurs, and it occurs frequently, that is really telltale. And we see here fever curves for 3 different malaria parasites, and you could even, if you're experienced, distinguish between the different malaria by the characteristics of the fever curve. We come back to the fever curve later, and then I explain why this is so weird, why the temperature fluctuates so much. And that has something to do with what the parasite does in infected people. Then we have anemia, and this is an indication that people lose their red blood cells. And then we have joint pain, we have splenomegaly. That is what this man is trying to find out here by touching the belly of that boy, is whether spleen and liver are enlarged. We have severe headache, vomiting, and coma. That is then when we have cerebral malaria, and that is often the cause of death, as it was in the case of my lecture. Then we have dark urine. That has, again, something to do with a lot of red blood cells. And malaria is normally a real killer for pregnant women and young children, less so for other people. But in areas and without treatment, pregnant women and children are the victims. So this slide is about cerebral malaria, and that's the vast majority of cases in children under the age of 5. So it's very tragic, it's very sad actually. And that is because protozoan proteins on the surface of infected red blood cells cause adherence to the internal lining of capillaries, blocking blood flow, causing hemorrhages, and then also impairing gene function, brain function. What we see here are several slides documented the accumulation of these cells in blood vessels and several stainings, so I will leave this for you to peruse at home. So this is, even if it looks slightly dodgy, I think it's very informative. This picture here I will come back to several times because it describes the life cycle of the Plasmodium. So I spared you largely the life cycle of all the other parasites before, but I really will be going on the life cycle of this one, and I can only encourage you to learn this one. All right, so we have here the mosquito landing on the arm of this person and is having a good time because it's drinking blood. So, and that's how it all starts. And if you don't like me explaining it, you can listen and watch movies explaining it all over the net. Some of them made in Australia, so WEHI is an abbreviation for an institute in Melbourne, and they make very good stuff, so check out the websites and have a look. So, okay, I already told you that the mosquito is female. It's not any mosquito. It has to be Anopheles mosquito. So this is the one that transmits the parasites. What it actually injects into people are sporozoites. They seem all a bit esoteric and special, but I really want you to learn this for malaria. The sporozoites, they look like a worm, so they have a worm-like appearance, but they're not a worm. So, and they get into the host, and then they will later become a different stage that then will go after the blood cells. These ones do not go after the blood cells straight away. They go to the liver first, and how they get there is shown here. So here we have the visual depiction of a sporozoite invasion of a blood vessel in the dermis. So the blue is the blood vessel, and in red here we see the trace of the sporozoite basically searching for the blood vessel and then homing in and then getting into that vessel. And it's green here because blue light on top of, or mixed with red light gives you green light. So what does this say? The blood vessel itself glows blue, and the sporozoite glows red, and here you see a trace. So the shutter has been open for some time, and you see where it all has been.
When it goes into the blood, the blood, does this occur inside the liver? Or
No. This occurs like a millimeter off the biting side. So the mosquito is a little dumb. The mosquito actually hits you more than once. It's trying to probe where the blood vessel is. And the mosquito also injects saliva into you, so to numb the action, so you do not instantly feel that you have been bitten, and to do all kinds of things. So there is very complex things going on between the mosquito and the person. And what it also does is, because the sporozoites, they live in the saliva glands of the mosquito, and if the saliva is injected into the host, then they get into the host, and they do not always get into the bloodstream, so they get around the bloodstream, and then they have to find their way into the bloodstream. Another question you could have asked is, why is this thing glowing? And that is glowing because people made it glow. So the sporozoites, they do not glow all by themselves. So they were giving a gene from another animal that made them glow. Here we have glowing sporozoites in a mosquito salivary gland. And this is a nice excuse to show these again. And this is, the diagram that I used to explain fluorescence in the last practical class, which I consider key, and I repeat this now, key to understand key technologies in immunology. Okay, but why do they glow? So what we see here is a jellyfish. So this is Equoria victoria. what a name. So it's a huge jellyfish. This actually here, it's not glowing. This is just a fancy photo against the sky through the water. But here is a jellyfish. And the jellyfish has been photographed under conditions where you see the bioluminescence. And the bioluminescence is constrained to cells in the periphery of the jellyfish. And that is the result of two proteins, aquarin and the famous green fluorescent protein. Aquarin binds calcium ions, and the protein undergoes a conformational change and oxidizes one particular group, which leads to the emission of blue light. So this one actually glows actively, but it glows in blue. But here, as you can see, the yellowfish glows in green. So how comes? A protein next to aquarin absorbs the blue light and then fluoresces in green light. Very similar to the fluorescent dyes we use in immunology to distinguish between T and B cells. Here, it's however not a dye, it is a protein. And this protein can absorb blue light and emit green light. So then some smart people said like, we take this protein out of context and put it into other organisms. And if you shine a UV light on them, then they will glow green. And they did this with mice. So this is a mouse, and this is a nude mouse without hair. That's why it looks so weird. But then you can see it better because you can see the skin without hair. And if you shine UV light on it, the whole mouse will glow. Okay, this is a kind of a folly a little bit, but it shows the principle. So they now made glowing rabbits and monkeys and everything, and if you really want to, you could even make people glow. So, but for us, and this is now scientifically a little bit more with a purpose, they made GFP, transgenic Plasmodium parasites. And then they can shine a UV light on a mosquito, and then they can see where the parasites are. So without cutting up the mosquito and trying otherwise to find out where the parasites are. And you can also see whether the parasites change location in a mosquito during their mental stage without killing the mosquito. So this is very neat to follow where the parasites go. And that eventually has been used to create this fancy photo to show how the sporocytes zoom in on a blood cell and then go. Okay, so now we have the sporocytes, after all this, inside the human body, and they go into the bloodstream and they eventually end up in the liver. So what do they do in the liver? So in the liver, they come here through the portal field, and then they will make contact with what is called a Kupffer cell, that's a resident immune cell in the liver, and then they use that Kupffer cell to get out of that vessel system here and into hepatocytes. And then you see these little red things here going from hepatocyte to hepatocyte to hepatocyte, and that's really weird. So normally if a virus infects a cell and it's in, then it can either replicate in the cell or not, it stays in the cell. These parasites, they go in the first hepatocytes, and then they don't like it, and then they go into the second, and then don't like it, and they go into the third. And this goes on and on and on until they finally settle into one, and then they multiply.
So they use the Kupffer cell to escape. Are you saying they're using it to escape the host blood cell?
No, in this case, what they do is, and this is again fluorescent sporocytes going from one hepatocyte to the other. So they make it very hard for the immune system to specifically find the infected hepatocytes because they left a trail of destruction behind, which looks like they are infected, but they're not because they've just been passing through. So the current understanding is for this, effort to go through several hepatocytes until you finally settle into one is as a decoy to hide the one that is truly infected. Okay, so now they're in the liver. And this is another depiction where the sporocytes go through the Kupffer cell, go to several hepatocytes, and then eventually multiply. And you can see there are many, many more cells that come out of an infected hepatocyte than those sporocytes that actually went into the liver. And they even do not completely destroy the hepatocytes, so they come out in what's called a merosome. So that's a bit of a hepatocyte, and there is still membrane enclosing the merozoites, and these are the parasites now, so they have transformed. So it's asexual multiplication, so one sporocyte makes many, many merozoites, but the merozoites look different. They are no longer worm-like. They're kind of roundish. And they stay inside the merosomes as long as possible, and if they eventually come out, and that could be far away from the liver, then they instantly track down a neighboring red blood cell and infect it. And then from then onwards, but let's have a look at how they get into the red blood cell first. So that infection takes only 15 to 20 seconds from the merozoite, and this is a merozoite here, kind of making contact with the red blood cell, kind of rolling up into the right position, using its organelles to induce the formation of a vesicle, and then staying inside that vesicle in the red blood cell and multiply, and eating it up alive, if you like. So they hide inside the red blood cell. So these are all electron microscopy pictures showing the multiplication of the merozoites in the red blood cell. So here's shortly after the infection, and this is shortly before the erythrocytes burst and the merozoites leave, and then they go after other red blood cells. So more electron microscopy images, and here is a blood smear with an infected red blood cell, and this is what the diagnostic will look like. So it's a very cheap way to diagnose it, is you look at the blood smear and you look for something inside red blood cells. They are normally kind of clear and semi-transparent, and they do not have discernible structures if you stain it like this. But if you see something, then you know it's a parasite, and it's a fair chance it's malaria. So this is further on in the developmental stage in that red blood cell. So here we get back to our overview. So I showed you that the sporozoites infect the liver. What comes out of an infected liver are merozoites. They infect red blood cells, and from then onwards, the multiplication is in red blood cells alone. It never goes back to the liver. And this is also a reason for the intermittent fever, because strangely, even days, even years after the infection, these things occur in a synchronized fashion. The release of merozoites from the red blood cells is synchronized. So they all release the merozoites at the same time. And that is the time where the fever peaks. And then you have another 5-6 days, whatever it is for the particular malarial species, and then that it takes to make more merozoites, and then they're all released at the same time again. And they do this because it's kind of a swarming technique. If you look at the war that's going on in Ukraine, so you can shoot down one drone, but you have trouble shooting 50 attacking you at the same time. And this is exactly what the malaria parasites do if they go from one red blood cell to the next. So they're synchronized, and that is the reason for the intermittent fever. Okay, so, but now we come back to, this is all asexual, to sexual multiplication. So some red blood cells, they actually form gametocytes, so male and female gametocytes. And for those to proceed, they need to be picked up by mosquitoes. And especially those transform the surface of the red blood cell by expressing proteins, and these make the red blood cell sticky, and then they get entangled in the lining of very narrow capillaries, which often happen at the next to the surface of the skin, and that increases the chance to be taken up by the next mosquito, because a mosquito is not going to drill 20 centimeters inside your body, so it's all a few millimeters at the surface. So this makes the malaria prone to cause, the effects in the brain. So it's all to increase the chance to get into the next mosquito, because that is needed, because the rest of the sexual reproduction has to happen in the mosquito, not the person. So we are only the intermittent host. It's not good for us because we may die, but we are only the intermittent host. So the main action, I mean the sex as far as Plasmodium is concerned, is happening in the mosquito, not in us. Okay, and this is a cross-section through the mosquito. So this is the head, here the salivary glands, and here's the main body. And here we have the fate of the malaria parasites inside the mosquito. So we have the female and the male gametocytes, we have the zygote, then it leaves the gastrointestinal tract, goes into the myxozoil, that is, they do not have a full blood vessel system, these insects, and through that they get to the salivary glands, these are supposed to be those, and then they can be injected through the mouth parts into the next host, into the next human being, if it's Plasmodium falciparum. Okay, so that is depicted here. So now we have like full circle, mosquito, sporozoites, liver, merozoites, red blood cells, gametocytes, mosquito, sexual reproduction, new host. So I tried my best to do the life cycle of Plasmodium in all its glory details. So now we come to the diagnosis. So there are many ways to diagnose malaria, but still the gold standard and still amazingly cheap, but requires some experience, is to prepare blood smear, take a microscope, and it could be a fairly cheap microscope. So the microscopes we use for teaching are perfectly fine for this. You don't need anything more fancy. And to look at your red blood cells. And then if you find infected cells, they will stand out. And then experienced people can even look at the infected cells, and depending on the appearance of the parasites in the red blood cell, are diagnosed with species of malaria it is. Okay, now we come to treatment and prevention. So famously, and this is also, as we see later, an example for bad science, is the whole connection with DDT. DDT is a very powerful insecticide, and this was appreciated so much early on that in 1948, two people, Paul Hermann Müller, or just one person actually, Paul Hermann Müller, a Swiss chemist, received the Nobel Prize for coming up with the idea. And DDT, it's dichlorodiphenyl trichloroethane, so this is a formula, kills insects by opening sodium ion channels in neurons, causing the neurons to fire spontaneously, so they get spasms and eventually they die. It was used extensively during World War II to control insect typhus and malaria vectors, so by both sides, so because they really cared about the fate about the soldiers. And that got rid of these diseases largely around the Mediterranean theater and also in other parts of Europe. So DDT was really helpful to get rid of malaria in Europe. So then some people got a little overboard here, and you see this is an advertisement poster from, I don't know what it's called, some producer or other from the US. So DDT is good for me, so they all scream. And maybe one person should not have done so. So this bird here may have waited a little bit, because as it turned out, there is some unexpected toxicity. And this is part of the Constitution of the US, and then you see all the stars, and here's a balded eagle, and they're very picky on this, and it was alleged that bald eagles, like many birds of prey, were especially affected by DDT due to biomagnification. This in itself is true. So DDT lingers around in the environment for relatively long periods of time, and it can aggregate up the food chain. So you try getting it into insects, and then you have things that eat insects, and then you have little birds that eat these carnivorous insects, and then you have bigger birds eating the small birds, and it goes up, up, up. And then it may accumulate in these top predators, and they may experience problems. For example, they may not be able to produce healthy eggs. So, and then we had this kind of hippie publication in the 80s, and that was Silent Spring. And they just thought like, oh, it is so quiet now, we have lost all the birds because of DDT. And that is not true. They have lost the birds of prey, perhaps to some degree of DDT, but as it turned out, more like through wild shooting than the actions of DDT. But DDT probably was never really be ideally used for agricultural purposes sprayed on the landscape by planes. This is not exactly how you would like to use it to fight malaria, for example. So, but whatever it was, in the 70s, DDT was banned by the United States, and the bald eagle population rebounded, which is a fine example of correlation is not necessarily causation. So this may have had a factor in this, but it wasn't the only factor. They also banned the hunting. So if people don't shoot the eagles out of the sky, this is obviously also good for the eagle population. So, but whatever it was, this kind of instituted the belief that DDT cannot be used under any circumstances, and it was banned worldwide. In the United States and other organizations tried really hard, so for other countries not to use it either. Which wasn't really that good for Africa, I think. So, before DDT became available, there were also methods to treat, to prevent malaria. One was removing or poisoning breeding grounds for mosquitoes by applying oil to places with standing water. I'm not saying this is environmentally friendly either, but you see the efforts that have been made early on to control malaria. So even if it goes to the extent of poisoning whole ecosystems. What now turns out to be really effective is the use of mosquito nets and bedclothes, and in particular mosquito nets that are treated with insecticides. So that the mosquitoes crawling around on the nets trying to find a hole or a fault or anything to get in will be dead before they find access. So, and that is, I think, as we speak now, the best way of doing it. Give people in affected area the means to protect themselves at the hours of the day where they are most exposed to Anopheles mosquitoes, and some species are actually specialized on biting people at certain times of the day. Vaccines have been developed, but they're not very efficient. So vaccines will not in the near future save the day. It's more these boring conventional methods that will do it for now. So then there are also options to treat malaria once you got it. And an old school drug is chloroquine. You will not use it today because most parasites are resistant due to the widespread use of it in the past. It's also not ideally the best drug because you have to apply it at doses very close to killing the host. And the mechanism of action is quite interesting and well investigated. That's why I go on a little bit on this. So inside the red blood cell, the malaria parasite degrades hemoglobin for the acquisition of essential acids. So during this process, the parasite produces a toxic and soluble molecule heme. That's the thing it cannot use. So it's the non-protein part of hemoglobin. To avoid destruction by this molecule, the parasite polymerizes heme to form the non-toxic hemozoin that collects in the digestive vacuole as insoluble crystals. Chloroquine enters the red blood cell by simple diffusion and becomes protonated in the acidic environment of the digestive vacuole, so it's a prodrug if you like, which keeps it in the vacuole. Chloroquine caps hemozoin molecules, which prevents further polymerization and subsequently results in cell lysis and parasite cell autodigestion. In essence, the infected cell drowns in its own metabolic products. So that is why it then preferentially damages the red blood cells with the parasites in it versus the other red blood cells. But there is a way out from the parasite's point of view. The parasite can pump chloroquine out of the digestive vacuole by specialized transporter proteins, and these are now found like throughout the world. So this is no longer useful, but it was a good drug at the time. So current malaria treatment is depending on doxycycline, and it's similar to tetracycline. And we had that when we talked its function as antibiotic to treat all kinds of bacteria. And it's against also the red blood cell stages of Plasmodium falciparum. So, and then there are other drugs, and there is a pipeline for develop drugs against malaria because there is also the tendency that the parasite can become resistant against all these drugs. So there was a recent Nobel Prize going back to 2015 awarded to a Chinese researcher for discovering artemisinin in a plant that grows in China. Okay, and the method of action of this drug is still unknown, but it does work. So, and it works so well, obviously, that the people thought it would be good to give the big prize for it. And again, as we had before when we talked about treating viruses, so the WHO strongly recommends the use of or using drugs in combination. And that always makes it harder for viruses, and in this case for parasites, to develop resistance, because they have to become resistant against several things at the same time, and the likelihood is reduced compared to you using these drugs in isolation. Okay, in the last couple of minutes, I would like to talk about naturally occurring resistance. So, in the areas that are affected by malaria, and in the past, as far as humans go, for most of the history they lived in Africa, and then only recently they marched out of Africa. So that the parasites that affect people and people that have a very long history on the African continent because of this. And so therefore there was also time for the humans to develop resistance, and one resistance is a mutation in the HBB gene, and that codes for one of our hemoglobin subunits. In the normal allele, this is often named HBA, in contrast to the sickle cell allele, HBS, has a mutation that changes one amino acid, glutamic acid valine, at position 6. And this change from a hydrophilic to a hydrophobic residue encourages binding between hemoglobin molecules, with the polymerization of hemoglobin deforming the red blood cell. So basically it makes the hemoglobin molecule sticky, and it impairs with its function, so this can flip into this. So this is normally not a mutation you want to have. So, and this happens, as far as I understand it, even more frequently if you climb up and go to higher altitudes. However, what it does is it makes you resistant to malaria. So people who are heterozygous with this mutation are still relatively resistant to malaria, but maybe will not win the next marathon race. If you're homozygous with the mutation, that is really not good for you at all. Then you may die even without the parasite around. So what you have here is that not even given infinite time, this allele will not reach 100% in a population, but it will linger around. There will be a lot of people heterozygous for it, and then on a population level provides an advantage in areas that are endemic for malaria, but not in areas that are not endemic for malaria. And therefore, distribution of this gene is more prevalent in areas that are affected by this parasite. There are other resistance, so for example, for a receptor, and this is sometimes called the Duffy antigen. So what you can see here again, that there is a host race between the parasite and the host, which resembles a little bit what happens between HIV and people. So again, you can have a little deletion in your receptor, and then you're almost completely resistant to HIV. So this happens, but it takes time and a lot of casualties. And so therefore, in the end, to have either a good treatment or even better, a vaccination would be highly desirable. Okay, and this is all I wanted to say. These are the learning outcomes. This is the last slide, and now I switch the lights on, and it's time for questions.
You said the mosquito bites you, injecting you with a sporocyte,
Mm-hmm.
right? And you also said the mosquito probes around for a bit trying to find a blood vessel.
Mm-hmm.
So if the mosquito is injecting the sporocyte into the blood vessel, then why does the sporocyte have to find the blood vessel again?
No, because the mosquito may bite you a few times without actually hitting the blood vessel in the first attempt, but still injecting saliva. Once the mosquito found the blood vessel, is not so much interested in injecting the saliva, it's just sucking up blood. So then the whole infection has already happened. That's how I understand it. Yeah. More questions? Again, more questions on prion and caliciviruses and whatever happened last week? Mm-mm. Okay, then we close a few minutes early, and I see you all in the practical classes very soon. No worries.